This is what it looks like. Whoops. There we go. This is a micro-injection needle, and you can take cancer cells and inject it. You can inject it into the heart, or you can inject it into some of the other blood vessels. In any case, that's how you get the that's where you can get the cancer into the metastases. Where do you get the cancer cells from? Cancer cells are a hugely used for all sorts of different studies. So even if you're not studying cancer, I use, for instance, cancer cell lines because they're really easy to take care of because they're very proliferative. So in other words, they're very commonly used in research, so it's really easy to get a hold of cancer cell lines. No, usually, well, like, collaborators maybe, but not like, you don't go to a cancer patient and say, hi, and, grab the cells. And I give this. Yeah. No, they're controlled lines, usually commercially distributed, and so that's where we get those from.
So you're doing just research, or are you developing treatments?
Well, in our lab, we're focusing predominantly on drug discovery. So we're really focused on finding new drugs, new compounds for the treatment of various diseases. So I was just discussing how we're looking for, we found actually some potential anti-cancer drugs by using the zebrafish as a model organism. So you can see this fish. You can see them under the microscope too. They're very beautiful little things.
And would you be looking for work in this kind of thing if you had the option of research, or would you be looking for?
I think so. Okay, these are 2-day-old zebrafish.
It's a large portion of what career I will go into.
But you can use this transgenic sequence. It has all blood labels for the recipe. So one really important process in cancer is the development of blood vessels. So tumors can't grow beyond a size of like 1 millimeter if they don't have their own supply of blood vessels. So inhibiting the growth of blood vessels in tumors inhibits tumors. So what we can do in zebrafish is before these blood vessels here develop, we can give it a compound and see whether that compound inhibits the growth of these blood vessels. And if it does, it has potential as an anti-cancer drug. It's very powerful. So imagine now screening 55,000 compounds. You're bound to find something. And we've actually found a number of potential novel anti-cancer drugs. One that's very interesting, an existing drug called, I think it's Lipitor, anti-cholesterol. To lower your cholesterol. We found that it has anti-cancer properties. So the really nice thing about that is it's much easier to get a drug approved for the treatment of something else if it's already approved in general, because that means it's already undergone extensive clinical testing and safety trials and all these things. So that's a rapid, right? And when you can do that with the zebrafish very rapidly and very quickly, you can get a lot of potential hits. Well, very important. You would think, but one thing that we kind of fail to appreciate is that how much we share in common with much of the life on Earth, right? So for instance, we learned a great deal about genes, genetics, and some of the genetic machinery involved from fruit flies. Fruit flies are very different from humans, but they share some of these very fundamental underlying mechanisms, right? So zebrafish are even closer to humans than fruit flies. Beyond, it does sound like a bit of a stretch, but while they do look different from humans, think about this, they do have a head, they do have a vertebrate, and so we can definitely learn much more from zebrafish than from fruit flies or worms or other model organisms. It doesn't. It becomes semantic when you look at, for instance, one of the projects I'm looking at now. I'm looking at iron metabolism and how that relates to neurodegenerative disorders like Alzheimer's and Parkinson's. And a very interesting, a new protein involved in iron metabolism was discovered in zebras. So we have it, and we discovered it in zebras. They're a very powerful little thing.
So how does iron metabolism?
Sorry?
How does iron metabolism affect Alzheimer's?
I'd be happy to tell you. So this is an emerging sort of area for research, which is one reason I'm excited to take on such a project. So iron is stored in specific proteins because if iron is just kind of let loose in the cell, it wreaks havoc. It's very reactive, you know. So it's toxic if it's not bound. But so as we get older, we get poorer at properly maintaining the proteins involved in iron storage, and that kind of leads to things like a larger amount of iron being stored in bodily tissue. Also, iron leaching into the cell, out of the protein into the cell and reaching and having toxic effects. So in Alzheimer's and Parkinson's and other neurodegenerative disorders, one thing that is common to all of them is you see a higher concentration of iron in brain tissue, right? So it's presumed that this is part of what causes, degenerative toxicity in the brain and whatnot. So what I'm working on now is I'm developing a new type of transgenic fish. So I'm genetically modifying a fish that will be able to look at compounds that are able to remove iron from the brain and from other parts of the body as well. So,
How about other metals?
Other metals?
Like such as mercury. You might remember the study done at the
My pleasure. Take care.
University of Calgary. They did a study on the effects of mercury, and there are also studies that show that increased amounts of aluminum also have an effect.
Yeah, that is true. The difference between iron and, let's say mercury or aluminum is that iron is necessary for the body. Think about red blood cells. There's iron in hemoglobin, which you need to have. It's part of the catalytic activity of the enzyme. So without iron, the enzyme wouldn't work. So in other words, because although it can be toxic, we have evolved a way to use it usefully for many different things. Iron is an essential cofactor for a wide variety of enzymes. So it's interesting because some anti-cancer drugs, actually, what they do is they remove iron. They remove iron from a specific enzyme called ribonucleotide reductase, which is an enzyme involved in DNA synthesis, right? And what are the cells that make DNA the most? It's going to be cancer cells, because they proliferate so rapidly, so they're always making DNA. So if you inhibit this enzyme that makes DNA, you inhibit the growth of these cancer cells. So iron is it's implicated in a lot of different mechanisms because it's a very useful, it's a very reactive atom. So mercury, for instance, to my knowledge, there is no enzyme that requires mercury to function. Mercury in any quantity is toxic. We have the equivalent of maybe one nail of iron in the totality of our bodies, whereas mercury, because we don't need it, there's going to be very little of it, and it's going to be toxic in any quantity, really, because we don't have the mechanisms to deal with it, right? You see other organisms, maybe they live in water pools under deep ocean vents where there's high concentration of of these otherwise toxic
Yeah.
compounds. And have a look. See if you can have a look. And you have, they have well-developed mechanisms for dealing with and using elements that are toxic to us. So I hope that answers your question then.
Yeah, no, I mean, I was referring to it as a toxin. It's a,
Yes.
it's responsible. The University of Calgary did a study where they determined that it was responsible for a lot of the symptoms.
Iron.
Mercury was
mercury, yeah.
responsible for the symptoms that are so characteristic of Alzheimer's and other types of dementia.
It's possible, certainly. I mean, these diseases are very complex, and it's a lot of different things that kind of converge and come together. But the role of iron in these disorders is not well understood. But there is evidence that removing the iron would have a potential positive therapeutic benefit. For instance, amyloid precursor protein is one of the proteins that's associated with plaques that form in neural tissue and displace, neurons and other things. Amyloid precursor protein is regulated by iron directly. So iron binds to an iron-responsive protein, which binds to an iron-responsive element on the amyloid precursor protein DNA or mRNA, and by that mechanism it controls its expression. So there's definitely something going on there, and it's worth a look to see what it is, right? So.
Yeah, I mean, of course you can't simply eliminate all iron from the body. You need the iron.
Iron is very, because we have evolved because we've evolved to use it in many different forms, it's very tightly regulated and tightly controlled. But as I mentioned, this sort of degrades with age. Although removing all iron would be toxic, would be deadly, removing an excess restores the normal amount of iron that someone might have. So,
I mean, I guess if you could remove it selectively from...
Exactly, and you can do that too. You see that different kinds of compounds, or different kinds of iron chelators, as they're called, are very selective. So for instance, that anti-cancer drug I was talking about, it only removes iron, it primarily removes iron from that one enzyme that has to do with DNA synthesis. So it's very specific, so it has a good therapeutic target, good therapeutic advantage potential. So, exciting stuff.
I'm not sure which one of the hospitals use a chelator for example, if someone receives multiple blood transfusions.
Yes. So that's another...
so there's a chelator that they use.
Lesser known, absolutely. That's actually where the idea for this project came from, is because there's little drug development for iron chelation in general, and the primary use for iron chelators today is for the treatment of, when you have somebody who has like a lifelong transfusion regimen. We have no normal way to excrete iron from our bodies, so it becomes deposited in very sensitive tissues like the heart and liver and brain. It can be very toxic, right? So for instance, beta thalassemia, sickle cell anemia, certain kinds of sickle cell anemia, and other similar disorders, that becomes a problem. And there's very little drug development with respect to that. There's only 2 drugs approved in Canada for that usage, and they have very narrow therapeutic ranges. So, for instance, they're both associated with autotoxicity, so a loss of hearing, which is irreversible and a serious side effect, right? So there are multiple reasons why we could benefit from having a better understanding of iron metabolism and, in particular, looking for novel iron chelators. So I'm very excited to get started on that. That's my summer project.
Oh, very good. So you're still in you're in
Undergrad.
University of Toronto.
That's right. I just finished my first year. So I'm studying neuroscience and computer science. And I'm like having a good time so far.
Good. Well, I better get running. I still want to catch the City TV as part of the tour. There's an hour left. Yeah. So nice to meet you.
Nice to meet you. I'm Philip.
Yeah. So I'm interested in. I mentioned blood transfusions. I'll tell you why. Because I don't know if you ever heard of let me see here. You ever heard of Jehovah's Witnesses, what they believe? Because, they don't accept blood products.
Yeah.
And actually, my dad was at St. Joseph's Health Center, and, the doctors didn't want to give him Venifer injections, which would have raised his hemoglobin. You can have that. And so, there was a big issue going on with that. They felt that there would be iron overload, but, the bone marrow aspiration test, they did it right after they gave him 3 consecutive injections. And they said, the slide shows that there's plenty of stainable iron.
Okay.
And I told him, I said, you took the test right after you gave him 3 injections. So don't you think that some of that iron from the injections ended up in the bone marrow? So, I had a very rough time with the doctors over that issue. Because, the fact is, a unit of blood has 250 milligrams of iron. So if the bone marrow is functioning normally, it is fully capable of producing hemoglobin at a very rapid rate. My dad, when they gave him 3 consecutive injections, iron injections, and his hemoglobin spiked 20 grams per liter in a couple of days. So, it shows that, it works, basically.
Well, that's wonderful. If it works.
Yeah. This is what I'm saying.
Besides, in addition to, because of religious restrictions, beliefs, in addition to that, besides that, there is a shortage, really, of blood, so it's in everybody's interest, really, to develop something that, because there's always also, there's sometimes a risk of, I'm not too sure, but I think there's sometimes a risk of rejection and other complications. And while that's a very tiny percentage, very tiny, it's still there. So you see generally a shift in medicine, personalized medicine, generation of tissue. Oh yeah, sure. Tissue from the patient's own cells, because there's less risk of immunorejection and better prognosis in general. Because there's very little supply, and there's a huge demand for things like organ transplant and blood transfusion. So it's in everyone's interest to be able to effectively utilize the most of the patient's resources, capabilities, shall we say.
Yeah, in fact, there's a doctor in this hospital, you might know of him, Dr. Friedman. He's the director of a program called OnTrack. It's the Ontario Transfusion Coordinators. And it's a government-operated program and operated by the Ministry of Health. And basically he did a whole research paper showing that not transfusing patients, but using non-blood alternatives, iron and what's the name of the drug that stimulates? I think it's called erythropoietin. Using those in combination, erythropoietin and iron is very effective in stimulating the bone marrow to produce red blood cells. And as you said, safer. So I mean,
it doesn't replace transfusional therapy for other reasons. So some patients might not be in a position to be able to produce their own blood, for instance. Wing, you want to use the cart?
No, I want to use that road note.
Okay, all right. So I'm afraid we need to start packing up.
I got to run. It's about 4:30, I think, right?